Somewhere in the cold, dark waters of the North Atlantic, a shark is moving right now. It is not in any hurry. It moves at less than 3 kilometers an hour, about 2 miles per hour, which is slower than most people walk. It has been moving like this, through water just above freezing, in near total darkness, for somewhere between 4 and 5 centuries. It was alive before the first European settlers crossed the Atlantic. It was alive before Isaac Newton described gravity. It may still be alive today, and no one knows exactly how old it is because science only worked out how to estimate its age in the last decade. That shark is real. It is called the Greenland shark, and it is only the beginning of this story. Off the coast of Iceland, a clam once sat on the seafloor for 507 years. It survived the Renaissance. It survived every war ever fought on the European continent. It survived the Industrial Revolution, the invention of the telephone, and the birth of aviation. It was alive when scientists finally pulled it from the water in 2006, and it died shortly after. Not from age, from the act of measuring it. Scientists froze it open to count the rings in its shell, the way you count rings in a tree trunk. Only then did they realize what they had done. In the depths of the Southern Ocean, an animal has been sitting in the same place since before the first cities were built, before the invention of writing, before the first pyramid was laid. It has spent 11,000 years quietly filtering seawater through a body made of living glass, growing less than a millimeter per year and showing no signs of stopping. And in the Mediterranean Sea, a jellyfish no larger than your fingernail has found something no other animal on Earth is known to do. When it ages, when its body begins to wear down and approach the end of its life, it reverses. It returns to an earlier form of itself and begins again. These animals are not myths. Every one of them has been confirmed and studied by modern science, and together they point toward a question that goes far beyond the ocean. What do they know about aging and death that we don't? Before we continue, if this is your kind of story, please take a moment to like this video and subscribe. It genuinely helps bring more of these to more people. And let me know in the comments, are you watching this late at night, or is your day only just beginning? Let's continue. There is a simple idea beneath every astonishing lifespan in this story, and everything else grows from it. Aging is not a physical law like gravity or the motion of the tides. It is a biological program. Most living creatures inherit that program, follow it from beginning to end, and never depart from it. The remarkable animals waiting farther below do not escape the rules of nature. They reveal that one particular rule can be written in more than one way. Every animal begins as a collection of cells, and every heartbeat, every movement, every wound that heals depends on those cells replacing older versions of themselves. A cell divides, then another divides after it, keeping tissues healthy and organs working. Yet each division leaves behind a minute trace of wear. Hidden within every chromosome is a protective structure called a telomere. It serves much the same purpose as the plastic tip on a shoelace. That small cap prevents the lace from unraveling into loose fibers. A telomere protects the end of a chromosome from fraying in much the same fashion, preserving the instructions carried within. The protection is not endless. Each time a cell divides, the telomere becomes just a little shorter. The change is almost unimaginably small, far too slight to notice in a single day or even a single year. Even so, countless divisions gradually trim away those protective ends. Eventually, they become too short to continue their task. Cells begin dividing less reliably, then stop altogether. Fresh tissue appears more slowly. Existing tissue becomes harder to maintain. Organs lose efficiency through accumulated years, and the body approaches the condition commonly called old age. This gradual decline is often mistaken for a disease, yet biology treats it differently. It is the expected conclusion of the program that governs most complex animals. The body is not suddenly forgetting how to function. It is following instructions that have existed since the beginning of its development, reaching the point where renewal gives way to gradual limitation. Even within that program, another layer exists. Every animal possesses an enzyme called telomerase. Its role is remarkably straightforward. It rebuilds telomeres, restoring part of what repeated cell division removes. During the earliest stages of life, this enzyme works vigorously, allowing rapid growth without exhausting those protective caps. In most adult animals, however, telomerase becomes almost entirely inactive. The machinery remains present, but its activity fades until only a small number of specialized cells continue using it. A similar pattern shapes another essential process. Every passing day exposes deoxyribonucleic acid to countless forms of damage. Ordinary metabolism creates reactive molecules. Natural radiation passes through living tissue. Chemical reactions leave behind tiny imperfections. Young bodies repair much of this harm with remarkable precision, correcting countless errors before they can spread. As the years accumulate, those repair systems become less dependable. Small mistakes remain. Additional mistakes join them. Gradually, the instructions inside cells become harder to preserve with complete accuracy, and the program moves steadily onward. The creatures gathered in this journey have each found a different response to that progression. Some continue using telomerase long after other animals have reduced it. Some divide their cells so slowly that telomeres lose almost nothing across immense spans of time. One astonishing species can even reset mature cells back to an earlier biological state, restoring its own life cycle instead of accepting its usual ending. Different bodies arrived at different solutions, yet all reach the same destination, a life that scarcely follows the familiar rhythm of aging. Before any of those strategies could be understood, scientists first needed proof that such extraordinary longevity truly existed. That proof emerged from an unexpected source, carried not by the ocean itself, but by traces left behind from humanity's nuclear age, preserved inside the eye of an ancient shark. Long before anyone could explain how this shark remained alive across centuries, scientists faced a simpler obstacle. They could not determine its age with confidence. The animal offered almost none of the biological landmarks that researchers normally rely upon, leaving one of the most remarkable lifespans on Earth hidden behind an absence of evidence rather than an absence of curiosity. The Greenland shark, known scientifically as Somniosus microcephalus, spends its life in the frigid waters of the Arctic Ocean and the North Atlantic Ocean. It commonly inhabits depths between 1,200 meters, which is about 3,937 feet, and 2,200 meters, which is about 7,218 feet. There, the water can fall to minus 1.8 degrees Celsius, or 29 degrees Fahrenheit, a temperature that slows nearly every process within its body. It is among the largest predatory fish in the world. A fully grown individual may reach 7 meters, that is nearly 23 feet, in length and weigh more than 1,000 kilograms, yet its growth is astonishingly gradual. On average, it adds only about 1 centimeter each year. A human lifetime can pass while the shark is still approaching full size, turning ordinary growth into a process measured not in decades, but in centuries. Most long-lived fish leave behind physical records of age. Tiny structures within the inner ear, called ear stones, accumulate annual layers much like the rings inside a tree. Some species also preserve age within hardened scales or other calcified tissues. The Greenland shark possesses none of these dependable archives. Its skeleton remains largely cartilaginous, and no reliable annual record forms inside its body. Throughout much of the 20th century, marine biologists suspected the species lived for an extraordinary length of time, yet suspicion alone could never become proof. The answer arrived from an event that unfolded far above the ocean. During the 1950s and the early 1960s, repeated testing of nuclear weapons in the atmosphere released immense amounts of radioactive carbon into the air. This heavier form of carbon entered plants, animals, and eventually the sea itself, becoming part of the global food web. By 1963, living tissues forming during that period carried a distinctive chemical signature. Researchers later named this worldwide marker the bomb pulse, an unintended timestamp written across the planet. The Greenland shark happened to preserve one remarkable part of its body, untouched from birth onward. The central lens of its eye forms before the shark is born. After that moment, no new material is added. Unlike skin, muscle, or blood, the lens remains chemically unchanged throughout the animal's entire life, preserving the carbon present when it first developed. Julius Nielsen and his colleagues at the University of Copenhagen recognized that this unusual feature offered something no other part of the shark could provide. They collected eye lenses from accidentally captured Greenland sharks and examined them using radiocarbon dating. The study included 28 individuals ranging widely in size. Smaller sharks clearly displayed the bomb pulse, revealing that they had been born after atmospheric testing had already altered the global carbon record. The largest sharks showed no such signal, meaning their eye lenses had formed before those radioactive traces entered the marine environment. From those measurements emerged one of the most extraordinary age estimates ever assigned to a vertebrate. The largest female, measuring a little more than 5 meters, which is about 16 feet, was estimated to be between 272 and 512 years old. The most likely age centered around 392 years, with an uncertainty of approximately 120 years, reflecting the limits of radiocarbon dating rather than any weakness in the evidence itself. When the results appeared in the journal Science during 2016, they established the Greenland shark as the longest-lived vertebrate ever confirmed. Even the bowhead whale, itself famous for exceptional longevity, falls far behind with a verified lifespan of about 211 years. Establishing that remarkable age solved one mystery, yet understanding how the shark maintained its body across such immense stretches of time required looking far deeper into its genetic blueprint, an investigation that finally arrived in 2024. The age estimate transformed the Greenland shark from an extraordinary curiosity into something far more valuable. Once researchers knew that a single vertebrate could persist across nearly 4 centuries, the next step was no longer counting years. It became an attempt to understand the instructions written into the animal itself, searching through its genetic blueprint for the mechanisms that had carried one body across spans of time usually reserved for civilizations rather than individual lives. 8 years after the landmark lifespan study, an international research team published the first complete sequencing of the Greenland shark genome. The result revealed one of the largest vertebrate genomes ever assembled. Hidden within that immense archive was not one dramatic mutation that explained everything, but an accumulation of subtle advantages, each reinforcing the next, like layers of maintenance, applied continuously over an exceptionally long existence. Among the most striking discoveries was an unusual abundance of genes involved in controlling inflammation and repairing deoxyribonucleic acid. In many vertebrates, including humans, these repair systems perform remarkably well during youth before gradually becoming less dependable as adulthood progresses. Tiny errors begin to escape correction. Damage accumulates molecule by molecule until cells lose efficiency and tissues slowly decline. The Greenland shark appears to follow a different pattern. Instead of relying upon a single copy of many essential repair genes, its genome contains several functional versions working together. These are not inactive fragments left behind by chance. They remain capable of performing their roles, creating overlapping systems that continue correcting damage even if one pathway becomes less effective. Rather than trusting a single repair crew to preserve the body, the shark carries several, each contributing to the same endless work. Researchers also found an extensive expansion of genes associated with ferritin, a family of proteins that safely stores iron within cells. Iron is essential for life, yet free iron can also drive destructive chemical reactions that generate highly reactive molecules. Left unchecked, these reactions gradually damage proteins, cell membranes, and deoxyribonucleic acid itself. In most animals, a small amount of this injury escapes repair each day, adding almost imperceptibly to the burden of age. The Greenland shark appears unusually well equipped to prevent that accumulation. Its expanded ferritin system captures excess iron before it can fuel widespread oxidative damage, reducing one of the steady sources of molecular wear that living cells experience throughout life. Combined with its extensive repair pathways, the result is a body that spends far more effort preserving itself than surrendering to gradual deterioration. Scientists at the University of Copenhagen have emphasized that these discoveries are not a blueprint for turning people into Greenland sharks. The practical ambition is much more restrained and far more realistic. By identifying the precise molecular pathways responsible for the shark's remarkable maintenance, researchers hope to design medicines capable of activating comparable pathways already present within the human body. The goal is not to replace human biology with something borrowed from another species, but to encourage existing repair systems to remain effective for longer than they naturally do. Seen from this perspective, the Greenland shark is not defeating time through determination or adaptation in any conscious sense. It is simply equipped with biological maintenance systems that continue working with exceptional reliability across centuries. Damage still occurs. Cells still experience stress. Molecules still break apart. Yet the balance remains tilted toward repair for far longer than it does in almost any other vertebrate known to science. Its solution is one expression of longevity, built upon patience, preservation, and relentless cellular maintenance. Elsewhere, beneath the ocean, another animal arrived at the same destination through an entirely different route, not by repairing itself more efficiently, but by enduring through a life measured in centuries, while resting almost motionless upon the seafloor near Iceland, where an unassuming clam began its life in the year 1499. Some discoveries begin with celebration. Others begin so ordinarily that no one present realizes history has already changed. The story of Ming belongs to the second kind, unfolding not through a dramatic expedition or a revolutionary invention, but through a routine scientific survey of the seafloor north of Iceland, where researchers were searching for the past climate of the North Atlantic rather than the oldest clam ever documented. In the year 2006, a team from Bangor University collected approximately 200 ocean quahog clams from the seabed. These shellfish were gathered as part of a project investigating long-term environmental change. Their shells preserve annual growth layers that allow scientists to reconstruct conditions from years long before modern instruments existed. Longevity was not the purpose of the expedition. The clams were expected to contribute information about the ocean, not about themselves. Among the collection rested one specimen that attracted no special attention. It was neither unusually large nor visually distinctive. Like the others, it was frozen and preserved before detailed examination could begin. Only afterward did researchers prepare the shell for age analysis, carefully counting the annual growth rings laid down across its lifetime. Each visible ring represented approximately 1 year of growth, formed gradually as the clam deposited new shell material along its edge. The process resembles the way a tree adds another ring to its trunk with each passing season, although the shell records life beneath the sea rather than life upon land. As the count continued, the numbers climbed beyond ordinary expectation, then beyond anything previously documented for a single animal of its kind. The final count reached 507 years. Working backward from that total placed the beginning of its life around the year 1499. Leonardo da Vinci was completing his anatomical studies. The Ottoman Empire stood near the height of its influence. European contact with the Americas was still only a few years old. While kingdoms shifted, ships crossed unfamiliar oceans, and ideas reshaped continents, this single clam settled into the seabed and continued the same patient rhythm of feeding and growing. The researchers eventually gave it the name Ming, honoring the Chinese dynasty ruling when the clam first began its life beneath the cold waters of the North Atlantic. Across more than 5 centuries, Ming endured the European Renaissance, the Reformation, the great voyages that connected distant continents, the Industrial Revolution, the abolition of slavery across many nations, 2 world wars, the nuclear age, and the emergence of the digital world. Generation after generation appeared and disappeared, while the shell added another thin layer, then another. Only after the rings had been counted did the researchers realize what had happened. The preservation process that protected the specimen for laboratory study had also ended the life of the oldest individual animal then known to science. No one involved had intended such an outcome. The extraordinary age remained hidden until the evidence was literally counted 1 year at a time. Members of the research team later spoke openly about the experience. One scientist described the event as among the most significant accidental losses in modern marine biology. The statement reflected more than regret over a remarkable lifespan. It acknowledged that a living organism carrying centuries of uninterrupted biological history had been ended before anyone recognized the rarity of what lay inside its shell. Even so, Ming did not leave science empty-handed. Every annual layer remained preserved, each one holding chemical traces of the surrounding seawater from the year it formed. Long after the clam itself ceased growing, the shell continued offering information no instrument designed by human hands had ever recorded continuously across more than 5 centuries. Its life ended in the laboratory, yet the archive it created across those centuries continues to reveal new chapters of the North Atlantic, one carefully measured ring at a time. A shell can become something far greater than a shell when time is allowed to settle upon it without interruption. Long after the living animal has ceased growing, the layers it created continue speaking through chemistry, preserving a history that no observer intended to record and no instrument could have maintained across the same immense span. Every year of an ocean quahog clam's life leaves behind another narrow band of shell. That layer forms from the surrounding seawater, incorporating tiny chemical signatures that reflect the conditions present at that particular moment. Water temperature influences growth. The availability of food leaves its own subtle mark. Salinity changes are preserved within the shell's structure, while variations in the chemistry of the surrounding sea become part of the material itself. The clam does not choose what to remember. Simply by remaining alive, it creates an uninterrupted environmental archive. Seen from that perspective, Ming was more than an exceptionally long-lived animal. It was a scientific instrument, operating continuously for more than 5 centuries without maintenance, adjustment, or calibration. Human technology has transformed the study of the oceans, yet even the most carefully designed monitoring systems remain limited by practical lifespans. Instruments are replaced, sensors drift, power supplies fail. Research programs eventually conclude. A single clam resting beneath the waters north of Iceland quietly accumulated year after year of information from the late 1400s until the year 2006, producing a record no manufactured device has ever matched in duration. Researchers from Bangor University examined those annual layers to reconstruct changes in the North Atlantic across 5 centuries. The shell revealed fluctuations in sea surface temperatures, preserving evidence from periods such as the Little Ice Age as well as the earliest stages of industrialization. Those centuries existed long before systematic ocean observations became possible, leaving relatively few continuous environmental records. Ming carried one of the rare exceptions, extending across generations that never imagined such information would someday be extracted from an ordinary shell. That is why the accidental loss of the clam carries scientific weight beyond simple sentiment. The shell survives, and its archive remains available for continued study, but the living organism that created it can never extend the record another year. The process ended the moment the clam was preserved. Every future season that might have become another carefully deposited layer vanished with it. The species itself continues to challenge familiar expectations about aging. Ocean quahog clams display what biologists describe as negligible senescence. As individuals become older, researchers do not observe the steady increase in natural mortality that characterizes most animals. A clam that has lived for 3 centuries is not measurably closer to death from aging than one that has lived for only 3 decades. Time passes, yet the ordinary biological program described earlier appears to advance with extraordinary restraint. Despite this remarkable biology, ocean quahog clams remain far from uncommon. They support major commercial fisheries throughout parts of the North Atlantic and are harvested in enormous numbers every year. They are among the principal shellfish used in the clam chowder traditionally served throughout New England. Marine scientists studying the species noted publicly during 2013 that many people who have enjoyed this familiar meal have almost certainly eaten animals already several centuries old, often without realizing that the shellfish on their plate had begun life long before the modern world emerged. Their extraordinary longevity exists beside ordinary human routines, largely unnoticed. They are harvested, sold, prepared, and eaten each day. They are neither legendary creatures nor carefully protected rarities. They simply persist beneath the sea, carrying centuries within shells that resemble countless others. Even so, remarkable as 507 years may be, the ocean has produced another solution to aging whose timescale stretches far beyond centuries, making even Ming's extraordinary life appear unexpectedly brief. There are moments when the scale of time becomes so immense that ordinary history begins to dissolve. Familiar landmarks disappear first, followed by remembered names, then entire civilizations, until only the slow passage of the planet itself remains. Within that vast span, one living creature has continued its existence with almost unimaginable persistence, rooted to the deep ocean floor from an age when the modern world had not yet begun. Its scientific name is Monorhaphis chuni. It is a glass sponge, anchored permanently to the seabed of the Indo-Pacific Ocean at depths reaching several thousand meters, that is nearly several miles below the surface. It does not hunt, migrate, or search for shelter. From the place where it first settled, it has remained attached throughout its entire life, filtering seawater that drifts endlessly through its delicate body. One confirmed individual has been estimated to be approximately 11,000 years old. When that sponge first began its life, the last ice age was drawing toward its close. Vast glaciers still shaped northern landscapes, and woolly mammoths continued wandering across parts of northern Europe and Siberia. Far to the west, the broad plain now hidden beneath the North Sea remained dry land. Today it is known as Doggerland, but at that time it supported Mesolithic hunter-gatherers who crossed ground that would later disappear beneath rising water. The sea had not yet claimed those valleys and plains that countless generations once called home. The sponge remained where it was, while coastlines transformed beyond recognition. Long before the first stones of Stonehenge were raised into the sky, it was already established upon the seabed. The pyramids of Egypt would not begin rising for another 6,000 years. Writing had not yet appeared. The wheel had not yet been invented. Agriculture itself remained young across much of the world, and cities belonged entirely to the future. Century after century accumulated without altering the sponge's patient existence. Kingdoms emerged and vanished. Empires expanded across continents before giving way to others. Dynasties succeeded one another. Languages were born, changed beyond recognition, and disappeared. Religions spread across oceans. Trade routes opened and faded. Borders shifted countless times. Throughout every chapter of recorded history, the sponge continued drawing seawater through its body, extracting microscopic nourishment with the same uncomplicated rhythm that had sustained it since prehistoric times. Unlike the remarkable animals encountered earlier, this lifespan extends beyond the reach of written memory itself. Human history becomes only a brief interval within the life of a single organism. Entire civilizations, from their earliest foundations to their final decline, occupy only portions of its continuous existence. Generations that regarded themselves as ancient are separated from the sponge's beginning by thousands of years still. It remains alive today, far beneath the sunlight. Surrounded by water that lingers near the freezing point, it endures beneath pressures that no unprotected human body could survive. Every moment, seawater passes through an intricate network of microscopic channels inside its body. Minute particles become nourishment. The water then returns to the surrounding ocean, and the process begins again without interruption. Across thousands of years, this modest exchange has continued almost unchanged, requiring no movement beyond the gentle current flowing through its living framework. Its persistence reshapes the meaning of longevity. The passing of centuries becomes almost insignificant when measured against a life extending back to the close of the Ice Age. Events that dominate history books become passing moments within a biological existence, still unfolding in the darkness below. Remarkable as that age may seem, the number alone does not explain the organism itself. The glass sponge achieves this extraordinary endurance because it is unlike almost every other animal on Earth. Its body is constructed according to principles so unusual that understanding its immense lifespan first requires understanding what, In the deepest biological sense, a glass sponge truly is. Some bodies survive by repairing themselves without pause. Others endure because almost nothing within them changes at all. The architecture of Monorhaphis chuni belongs to the second path, built upon a design so unlike most animal life that familiar comparisons gradually lose their meaning. It has no brain-directing behavior, no heart circulating blood, no blood itself, no nervous system carrying electrical signals, and no digestive system resembling those found in more familiar creatures. Even so, it remains unmistakably alive. The entire organism is organized around a remarkable internal structure known as the basal spicule. This slender rod is composed of biologically produced silica, the same fundamental material found in glass, and it can reach approximately 3 meters in length, that is nearly 10 feet. Anchored deep within the seafloor, the spicule supports the sponge throughout its existence, providing both stability and structure, while surrounding tissues gradually expand around it over thousands of years. The body built upon this framework follows an equally unusual design. In most animals, tissues consist of countless individual cells separated by delicate boundaries. Each cell performs its own tasks while communicating with its neighbors. Monorhaphis chuni abandons much of that arrangement. Large portions of its living tissue are syncytial, meaning the cells have fused into one continuous network that shares the same internal fluid. Nutrients, chemical signals, and cellular resources move across this connected system without encountering the countless barriers present in ordinary tissues. That organization changes the way damage is handled. When stress affects one region of the sponge, the surrounding network can redistribute resources across the continuous living structure rather than relying upon isolated cellular responses. Wear becomes a shared burden rather than a local crisis. Instead of many independent compartments struggling separately, much of the organism functions as one integrated living fabric extending across its entire body. The basal spicule itself also records the passage of immense stretches of time. Throughout the sponge's life, microscopic layers of silica are deposited one upon another, forming delicate concentric bands that slowly thicken the structure. The process resembles the annual growth of a tree, only in the broadest sense. Here, the material is glass rather than wood, and the record extends not across centuries alone, but across many millennia. Every thin layer preserves chemical information from the seawater surrounding the sponge when that portion of the spicule formed. Scientists examining these layers have reconstructed environmental history reaching back toward the close of the last great glacial period. Tiny variations in the proportions of magnesium and calcium preserved within the silica reveal changes in ocean conditions over extraordinary spans of time. Isotopic signatures record episodes associated with hydrothermal activity, while broader chemical patterns preserve evidence of long-term temperature shifts as the planet emerged from the final stages of widespread glaciation. The sponge never intended to create this archive. It simply continued growing, allowing the chemistry of each passing age to settle permanently into its skeleton. Its longevity also depends upon remarkable restraint. The sponge consumes very little energy because it performs very little movement. Cold, deep water slows the chemical reactions that normally contribute to biological wear. The silica framework resists deterioration far more effectively than soft organic structures alone, and the surrounding environment changes only gradually across the passing seasons. Powerful storms never disturb the depths where it lives. Food shortages remain comparatively uncommon because microscopic particles drift continuously through the water. Predators capable of destroying such a large, firmly anchored sponge are uncommon. The result is a life measured not through activity, but through persistence. Monorhaphis chuni remains where it first settled, allowing stability itself to become a strategy for enduring almost unimaginable spans of time. Yet the deep ocean also produced another solution entirely, one that abandoned endurance in favor of renewal, replacing steady persistence with the astonishing ability to begin life again from the beginning. Among all the different ways life has negotiated with time, perhaps none appears to bend the ordinary rules more completely than a creature small enough to rest upon the tip of a finger. It drifts through coastal waters almost unnoticed, transparent enough to disappear against the surrounding sea. Yet within its delicate body exists one of the most remarkable biological abilities ever documented. Its scientific name is Turritopsis dohrnii, commonly known as the immortal jellyfish. Fully grown, the adult measures about 4.5 millimeters across, which is about 0.18 inches. Its bell is nearly transparent, allowing a vivid red stomach to remain visible through the surrounding tissue. Around the edge of the bell, tentacles increase in number as the animal matures, beginning with only a handful in youth and eventually reaching as many as 90 in adulthood. Its life begins much like that of many other jellyfish. A tiny larva drifts through the water before attaching itself to a suitable surface on the sea floor. There it transforms into a small polyp, a simple stationary form that resembles a miniature branching tube rather than the graceful swimmer most people imagine when hearing the word jellyfish. From this polyp, new medusae gradually bud outward, separating from their parent before swimming freely through the surrounding ocean as mature adults capable of reproduction. For almost every other jellyfish species, adulthood follows a familiar biological path. Reproduction is succeeded by gradual deterioration. Cells accumulate damage, tissues weaken, and the body eventually reaches the endpoint toward which its life cycle was always directed. Aging completes the program written into its biology. Turritopsis dohrnii follows that path only until conditions become unfavorable. When exposed to severe physical injury, prolonged starvation, environmental stress, or advanced age, the mature medusa begins an astonishing transformation. Rather than continuing toward death, its bell slowly folds inward. The tentacles shorten, then are gradually reabsorbed into the body. The familiar umbrella-shaped form collapses upon itself until almost none of the original anatomy remains recognizable. Attached to a nearby surface, the animal becomes a small rounded cyst, appearing less like a jellyfish than an unremarkable patch of living tissue. Across roughly 48 to 72 hours, a remarkable reorganization unfolds within that simple structure. The cyst gradually develops into a juvenile polyp, returning to precisely the stage occupied near the beginning of its life. Once established, the polyp resumes ordinary growth, eventually producing entirely new adult medusae through budding. The individual has not produced offspring while remaining unchanged. Instead, the adult itself has reversed its own developmental history, returning to youth before beginning the cycle again. No confirmed biological limit has yet been identified for how many times this reversal may occur. Under suitable laboratory conditions, researchers have repeatedly observed the process taking place again and again. In principle, the cycle may continue indefinitely, provided disease, predation, or catastrophic injury does not intervene. The cellular mechanism responsible is known as transdifferentiation. In this process, a fully mature, specialized cell abandons the identity it has maintained throughout its working life and becomes an entirely different kind of specialized cell. A muscle cell can become a nerve cell. A skin cell can become a reproductive cell. Instead of remaining permanently committed to one role, the cell rewrites its biological identity, exchanging one function for another as development begins anew. Most animals lose this flexibility soon after early development. Their cells settle into permanent occupations that remain fixed for life. Turritopsis dohrnii retains an extraordinary ability to reverse that commitment, allowing mature tissues to rebuild an earlier version of the organism rather than simply maintaining the existing one. In doing so, it does not merely postpone aging, it returns to the opening chapter of its own biological story through a process that has become one of the most ambitious goals in modern regenerative medicine, where researchers continue searching for ways to reproduce artificially what this tiny jellyfish accomplishes naturally. Sometimes, the greatest scientific revolutions begin with a discovery that changes how researchers think about life itself. A single experiment can overturn assumptions that seemed permanent, revealing possibilities that had remained hidden beneath ordinary biology. Such a moment arrived during the early years of the 21st century, when an unexpected breakthrough transformed the study of aging, regeneration, and the remarkable flexibility still concealed within mature cells. In the year 2006, Japanese researcher Shinya Yamanaka announced a discovery that reshaped modern biology. He demonstrated that fully differentiated adult human cells could be reprogrammed into a pluripotent state, one in which they regained the ability to develop into many different kinds of specialized tissue. By activating a small combination of carefully identified genes, mature cells that had long been committed to a single identity could effectively return to an earlier developmental condition. The scientific community recognized the profound importance of this work, and it ultimately earned the Nobel Prize in Physiology or Medicine. The genes responsible for this transformation became known collectively as the Yamanaka factors. Their discovery opened a path towards stem cell therapies without requiring the destruction of embryos, creating new possibilities for studying disease and exploring future regenerative treatments. Equally important, the work challenged the long-held belief that cellular aging always moved in only one direction. Mature cells, it seemed, possessed the capacity to recover developmental potential once thought permanently lost. Yet while laboratories celebrated this achievement, another organism had already been performing an even broader version of the same biological feat for countless generations beneath the sea. The tiny jellyfish encountered earlier does not rely upon specialized equipment, carefully measured laboratory chemicals, or precisely controlled experimental conditions. When exposed to severe stress, it begins reorganizing its entire body. Within approximately 72 hours, the adult form returns to its juvenile stage, reversing development through a coordinated process that transforms mature tissues into entirely new structures. What researchers accomplished cell by cell under carefully managed conditions, the jellyfish accomplishes throughout its whole body as part of its ordinary biology. As scientists examined this remarkable process more closely, they sequenced the transcriptome of Turritopsis dohrnii. A transcriptome represents the complete record of which genes are active at a particular stage of an organism's life. Rather than showing every available genetic instruction, it reveals the instructions currently being used. During the reversal process, investigators found that the jellyfish activates genes associated with the same fundamental pathways involved in restoring pluripotency, including factors closely related to those identified through Yamanaka's pioneering work. The similarity immediately attracted worldwide attention. The jellyfish does not imitate laboratory methods. Instead, it appears to reach a comparable biological destination using its own naturally evolved genetic program. Stress alone initiates the sequence. Once begun, the transformation unfolds through carefully coordinated changes in gene activity until a new juvenile polyp emerges from tissues that had previously belonged to a mature medusa. Research groups across Spain, Italy, Japan, and the United States continue examining this sequence in extraordinary detail. Their goal is not merely to observe the transformation, but to identify the precise molecular signals that permit complete cellular reprogramming within a living animal while maintaining the integrity of the organism as a whole. Each newly identified pathway provides another clue to mechanisms that conventional biology once considered impossible. The medical possibilities under investigation extend across several fields. Scientists are exploring whether similar principles might someday encourage replacement of neurons lost during Parkinson's disease, regeneration of cardiac muscle damaged after heart attacks, or repair of the cellular injury underlying multiple sclerosis. Those ambitions remain subjects of active research rather than established therapies, yet they demonstrate how one tiny marine animal has reshaped scientific thinking. The jellyfish accomplished all of this without laboratories, instruments, or awareness that its biology would someday inspire one of the most ambitious areas of modern medicine. For more than a century after its discovery, its extraordinary ability remained visible to science, patiently awaiting the moment when human understanding finally caught up with what nature had already accomplished. Discovery does not always arrive at the moment something is first seen. Sometimes an organism enters the scientific record, receives a name, occupies shelves in museums, appears in research notes, and waits patiently for generations before anyone recognizes what it has been demonstrating all along. Knowledge often advances not because nature changes, but because human understanding finally reaches the point where familiar observations acquire entirely new meaning. The small jellyfish known today as Turritopsis dohrnii entered science during the late 19th century. In the year 1883, the German naturalist Friedrich Waismann collected and formally described specimens from the Mediterranean Sea. At the time, the animal appeared to be an ordinary member of the hydrozoans, a group already containing many small and seemingly similar species. Nothing about its appearance suggested that it possessed one of the most extraordinary biological abilities ever documented. Once cataloged, the species settled into scientific collections much as countless others had done before it. Specimens were preserved in museum jars. Living individuals appeared in research aquariums. Marine biologists recorded their distribution in species inventories and taxonomic studies. Across more than a century of observation, the jellyfish remained present before researchers without revealing what it was truly capable of accomplishing. The remarkable reversal described earlier unfolded unnoticed because no one expected such a process to exist. The turning point arrived not through a deliberate search for biological immortality, but through careful observation of ordinary animal behavior. During the late 1980s, a marine biology student named Christian Summer studied hydrozoans near the Italian coastal town of Rapallo. His work focused upon understanding the daily lives of these delicate drifting animals rather than searching for unprecedented mechanisms of longevity. As his observations continued, something unexpected emerged. Aging and stressed specimens of Turritopsis dohrnii were not following the familiar sequence observed in other jellyfish. Instead of progressing steadily toward death, they appeared to begin transforming into an earlier developmental stage. At first, the phenomenon seemed so unusual that it demanded repeated confirmation. Sommer spent many months documenting the process carefully, ensuring that what he observed represented genuine biology rather than accident or error. Those observations eventually formed the foundation of the first scientific publication describing the remarkable reversal, which appeared during the year 1996. Despite the extraordinary implications, the study attracted relatively little attention. The wider biological community acknowledged the report, yet few researchers immediately recognized the significance of what had been described. The discovery remained largely confined to specialists studying marine invertebrates, distant from the central questions then shaping modern biomedical research. Only during the early years of the 21st century did circumstances begin to change. Stem cell research expanded rapidly, and scientists increasingly explored the possibility that mature cells might recover developmental flexibility once considered permanently lost. Cellular reprogramming became one of the defining questions in biology. As these ideas matured, researchers returned to the tiny jellyfish with an entirely different perspective. They were no longer asking whether the observations had been accurate. They were asking how such a transformation could possibly occur. With that conceptual shift, a century of overlooked evidence suddenly acquired profound importance. The organism itself had remained unchanged throughout the intervening decades. Museum specimens, laboratory colonies, and published descriptions had all existed in plain view. What had been missing was not access to the animal, but the intellectual framework required to interpret its behavior correctly. Nature had offered the same demonstration repeatedly, while scientific understanding gradually evolved toward the ability to recognize it. The story reveals an unusual partnership between discovery and readiness. An organism capable of reversing its own life cycle rested within scientific collections for more than 110 years before biology possessed the ideas necessary to appreciate what it had quietly been revealing. The ocean had never concealed the phenomenon. Human knowledge simply required time to grow into the question that finally allowed the answer to become visible. Not every path away from aging depends upon extraordinary repair or the ability to begin life again. Some organisms approach the problem by quietly reshaping the very idea of what it means to remain the same individual through time. In their world, continuity is not preserved by keeping one body intact, but by allowing life itself to divide without bringing existence to an end. Sea anemones belong to this unusual category. Fixed to rocks, shells, or the seafloor, they often appear unchanged across passing seasons, extending rings of soft tentacles into the surrounding water as currents deliver microscopic prey. Beneath that familiar appearance, however, lies a method of persistence unlike that of almost any other animal discussed so far. One of their most common forms of reproduction is clonal fragmentation. Small portions of the body separate from the parent and continue growing independently. Each fragment carries the same genetic information as the original animal, eventually developing into a complete sea anemone indistinguishable from the one from which it came. The lineage expands without requiring the combination of genetic material from another individual. Some species perform an even more remarkable act. Through a process known as binary fission, a single sea anemone slowly divides down the middle of its own body. The split proceeds lengthwise until two complete animals remain, where only one existed before. Neither half represents damaged tissue struggling to recover. Both halves continue living as fully functional organisms, complete with mouths, tentacles, and internal structures. The original animal has not perished. it has become two living continuations of itself. Those two anemones may eventually divide again, and the process can continue repeatedly across generations. The genetic lineage extends forward without any obvious biological terminus. Rather than following a single uninterrupted body through time, persistence emerges through continual branching, each division carrying the same inherited identity into multiple living descendants that are genetically identical to the organism from which they emerged. Sea anemones also display what biologists describe as negligible senescence. Unlike the exceptionally old clam and Greenland shark encountered earlier, which represent unusually aged individuals belonging to species that still possess finite lives, sea anemones, as a broader group, show no measurable increase in mortality rate simply because they become older. As years accumulate, the probability of dying during any particular year does not steadily rise in the way observed across most animals. Age alone appears to impose remarkably little additional risk. Some genetic lineages formed through repeated division are estimated to have persisted for thousands of years. Establishing the exact age of such colonies presents considerable scientific difficulty because the bodies composing them have continually divided, regenerated, and replaced earlier forms. Unlike counting annual growth layers within a shell or reading chemical records preserved inside a glass skeleton, no straightforward biological calendar remains intact. Researchers instead reconstruct histories through genetic relationships, patterns of growth, and long-term ecological observation, recognizing that the lineage itself has continued even while individual bodies have repeatedly changed. This persistence introduces an unusual philosophical challenge. Imagine watching a sea anemone divide into two complete animals, both surviving without interruption. Which one should be considered the original individual? The answer resists ordinary definitions because neither animal possesses a stronger claim than the other. Both arise directly from the same uninterrupted living body. Identity ceases to belong exclusively to one organism and instead becomes shared across two equally continuous lives. In that sense, death no longer functions as a destination written into an internal biological program. For sea anemones, endings generally arrive through external events such as predation, disease, environmental disruption, or the destruction of habitat. Remove those outside forces, and available evidence suggests no fixed biological timetable demanding that life must eventually conclude. Their remarkable persistence does not eliminate mortality altogether, but it dissolves many of the boundaries that usually separate one life from the next, allowing continuity itself to become the enduring feature rather than any single body. Far below the changing surface, where the passing centuries leave almost no visible signature upon the surrounding water, there are colonies that measure existence with extraordinary patience. Their history is not written through movement or dramatic transformation. It accumulates so gradually that even an entire human lifetime is scarcely enough to witness more than the smallest fraction of their growth. During the month of March in the year 2009, researchers from the National Oceanic and Atmospheric Administration explored remote deep waters within the Papahanaumokuakea Marine National Monument, northwest of the Hawaiian Islands. There they encountered an ancient colony of black coral, unlike any previously documented. Careful age analysis placed the colony at approximately 4,265 years old, making it one of the oldest known living marine animals ever examined. Its name can be misleading. Black coral is neither a plant nor a mineral formation. It is an animal belonging to a group of colonial organisms composed of countless tiny polyps. Each polyp is a living individual, yet all remain physically connected, sharing a common skeletal framework that supports the entire colony. Rather than existing as isolated animals gathered together by chance, they function collectively as one integrated living structure, extending outward from a shared foundation. That framework develops through remarkable persistence. Each polyp secretes thin layers of a protein-based skeleton, gradually strengthening the colony while providing support for future growth. These layers accumulate one upon another with extraordinary slowness. In some deep water species, expansion may proceed at less than 1 millimeter each year, which is about 0.04 inches. The difference from one year to the next is almost impossible for a person to perceive directly. Only the patient accumulation of thousands of years reveals the true scale of the colony's existence. When this particular coral first began growing, the Great Pyramid of Giza was still rising above the desert of ancient Egypt. Across the eastern Mediterranean and the Near East, Bronze Age societies continued refining metallurgy, trade, and organized cities. Systems of writing had existed for only about 500 years, preserving human memory through symbols newly committed to clay and stone. Since that distant beginning, the colony has continued adding one microscopic layer after another, preserving its own uninterrupted existence while civilizations appeared, transformed, and disappeared beyond the reach of the deep ocean. For almost all of that immense history, no human being ever observed it. The colony occupied depths inaccessible to divers, remaining beyond direct exploration until the development of modern deep-sea submersibles during the 20th century. Its world remained physically separated from ordinary human experience, not because it was hidden deliberately, but because the technology required to reach it had not yet been invented. The same depth that delayed discovery also protected the colony. Many of the hazards that threaten shallow water corals scarcely reach these remote environments. Powerful storms expend their energy far above. Anchor damage never arrives. Crown of thorns, starfish, capable of devastating tropical reefs, rarely inhabit such depths. Even the widespread bleaching events associated with warming surface waters exert far less influence upon these ancient communities. Their surroundings remain comparatively stable across long stretches of time, providing conditions that allow extraordinarily gradual growth to continue without frequent interruption. That stability does not eliminate every danger, geological disturbance, changing ocean chemistry, or human harvesting can still threaten deep water corals. Yet, compared with their shallow relatives, these colonies exist within an environment where destructive forces arrive far less often. Longevity emerges not from rapid repair or dramatic renewal, but from uninterrupted persistence, each nearly invisible layer, preserving another year of existence until thousands of years become embodied within a single living structure. Across the ancient shark, the enduring clam, the glass sponge, the remarkable jellyfish, the ever-dividing anemone, and this patient coral, the pathways toward extraordinary longevity appear strikingly different. Yet beneath those different bodies and different histories lies a deeper biological pattern, one that reaches beyond individual species and begins to reshape the very foundations of how aging itself is understood. Across every remarkable creature encountered so far, the outward differences are impossible to ignore. One carries a skeleton of glass, another drifts as a transparent jellyfish. One grows as a colony of countless polyps, while another glides through Arctic waters with the slow confidence of an ancient predator. Their bodies follow entirely different blueprints, yet beneath those visible differences, a quieter pattern begins to emerge. It exists, not in shape or behavior, but within the molecules responsible for preserving life itself. One of the most important participants in that pattern is telomerase, an enzyme found in every known animal. Its role is to rebuild the protective caps located at the ends of chromosomes. Those protective regions help preserve genetic information each time a cell divides. During the earliest stages of development, telomerase remains highly active because rapidly growing tissues require continual protection against the gradual shortening of chromosome ends. For most animals, however, that activity changes dramatically after development is complete. In adulthood, telomerase becomes greatly reduced throughout the majority of ordinary body tissues. Only reproductive cells and certain populations of stem cells continue maintaining substantial activity. As years pass and cells divide repeatedly, chromosome ends gradually lose protection, contributing to the accumulated cellular wear associated with aging. The long-lived marine animals explored throughout this journey follow a different pattern. Studies suggest that ocean quahog clams, Greenland sharks, glass sponges, and the immortal jellyfish all maintain telomerase activity within ordinary body tissues at levels uncommon among most adult animals. These are somatic cells, the regular cells that build muscles, organs, connective tissues, and countless other structures unrelated to reproduction. Rather than abandoning chromosome maintenance after early development, these species continue protecting their genetic material throughout life. This shared feature appears despite the extraordinary differences separating their evolutionary histories. The clam did not inherit its longevity from the shark, nor did the jellyfish borrow its biology from the sponge. Each lineage arrived independently at a similar principle. If genetic information must remain reliable across centuries or even millennia, the ends of chromosomes cannot simply be left to gradual deterioration. They require continual maintenance long after ordinary development has ended. Protection alone, however, is not enough. Every living cell also experiences countless forms of genetic damage arising from ordinary metabolism, naturally occurring radiation, and chemical reactions unfolding throughout life. Deoxyribonucleic acid repair systems continually identify these injuries, replacing damaged sections before errors accumulate beyond recovery. In most species, those repair systems gradually become less effective as age advances. Among the longest-lived marine animals, the opposite pattern appears to dominate. Each species accomplishes this objective through its own biological design. The Greenland shark possesses expanded collections of genes associated with repairing damaged deoxyribonucleic acid, creating additional layers of protection across immense spans of time. The glass sponge distributes injury across its continuous syncytial tissues, preventing localized damage from overwhelming isolated regions. The immortal jellyfish periodically reorganizes its entire body through cellular reprogramming, restoring mature tissues to an earlier developmental condition where repair mechanisms begin operating anew. 3 organisms, separated by vast evolutionary distances, reveal 3 distinct engineering solutions. None copies the others directly, yet all converge upon the same underlying requirement. The genome must remain dependable for as long as the organism continues living. Without preserving genetic instructions, no amount of slow growth, remarkable regeneration, or extraordinary longevity could endure. Maintaining such extensive protection demands energy. For many animals, especially those living active lives within constantly changing environments, the metabolic expense may outweigh the potential benefit. Every additional repair process consumes resources that could instead support rapid growth, reproduction, or escape from predators. Evolution, therefore, balances maintenance against immediate survival rather than pursuing unlimited longevity. Within the deep ocean, that balance changes. Slower metabolism reduces the pace of accumulated damage, while comparatively stable surroundings lessen many external stresses that constantly challenge life nearer the surface. Under those conditions, investing energy in preserving the genome across extraordinary spans of time becomes a far more successful strategy, allowing biological possibilities that would remain difficult to sustain in the restless waters above. Longevity is often imagined as a property carried entirely within an organism, encoded inside its genes and preserved by its cells. Yet every living thing exists within an environment that shapes what those genes can accomplish. A body does not age in isolation. It ages while continually responding to the world around it, spending energy to repair damage, resist change, and survive each passing day. In the deepest regions of the ocean, many of those demands become remarkably different. Among the most important influences is temperature. Every chemical reaction inside a living cell proceeds at a rate influenced by warmth or cold. Reactions that release energy, copy genetic material, repair damaged proteins, and prepare cells for division all slow as temperatures fall. For organisms adapted to these conditions, cold does not merely reduce activity, it fundamentally alters the pace at which life unfolds. The Greenland shark offers one of the clearest examples. It spends much of its life in waters approaching minus 1.8 degrees Celsius or 29 degrees Fahrenheit. At those temperatures, its metabolism operates with extraordinary restraint. Cells divide far less frequently than those of warm-blooded mammals, whose bodies maintain much higher internal temperatures. Every division carries the possibility of accumulating small imperfections and gradually shortening chromosome ends. By dividing less often across each passing year, the shark reduces one of the central processes associated with biological aging. The surrounding water performs part of that work simply by remaining cold. Light also disappears with depth. Beyond approximately 200 meters, which is about 656 feet, sunlight fades until the familiar brightness of the surface no longer reaches the seafloor. Among the many consequences of that darkness is protection from ultraviolet radiation. Near the surface, ultraviolet light continually damages deoxyribonucleic acid, producing strand breaks and chemical alterations that cells must constantly repair. Deep-sea organisms avoid much of this particular burden because the radiation responsible never penetrates to their world. The environment also remains unusually stable. Surface ecosystems experience changing seasons, powerful storms, fluctuating temperatures, and shifting supplies of food. Many animals must respond by migrating, reproducing rapidly, escaping predators through sudden bursts of movement, or enduring prolonged periods of scarcity. Every one of those responses demands energy that cannot simultaneously be invested in maintaining tissues and preserving cellular integrity. Conditions in the deep ocean differ profoundly. Temperatures vary only slightly across immense stretches of time. Powerful waves never disturb the seafloor. Day and night lose much of their meaning where sunlight cannot reach. Many deep-sea predators move deliberately, and countless organisms survive through slow, carefully balanced metabolisms rather than constant competition. Under such circumstances, the biological budget changes. Resources that might otherwise be spent responding to continual environmental upheaval can instead support the ongoing maintenance of cells, proteins, and genetic material. Even the immense pressure becomes part of this remarkable setting. Although such pressure would rapidly prove fatal to an unprotected human body, deep sea animals evolved within it across millions of years. Their proteins, cell membranes, and internal chemistry function naturally under conditions that isolate them from many of the biological pressures experienced closer to the surface. The crushing weight of the ocean does not hinder their lives. Instead, it forms another boundary, separating their world from the restless conditions above. Taken together, these environmental features reveal that the deep ocean did not accidentally become home to many of the longest-lived animals on Earth. Cold temperatures, reduced radiation, extraordinary stability, and specialized adaptation collectively created circumstances in which maintaining the body across centuries became an evolutionary advantage. Over immense spans of time, life responded by producing organisms capable of sustaining themselves with extraordinary persistence. Yet persistence is not the same as invulnerability. Every remarkable creature encountered throughout this journey remains vulnerable to disease, predation, environmental disruption, or physical destruction. Their biology may postpone the ordinary processes of aging to astonishing degrees, but it cannot remove the reality that existence still depends upon surviving the world around them. The word immortality carries an impression of permanence, as though time itself has surrendered its influence. Yet the creatures gathered throughout this journey reveal a far more delicate truth. Their extraordinary biology does not place them beyond harm. It changes only their relationship with aging. The world around them continues to hold countless ways for a long life to end in a single moment. The immortal jellyfish offers perhaps the clearest example of this distinction. Its remarkable ability to reverse its own life cycle protects it from the gradual decline normally associated with growing older. It does not protect it from hungry predators. Fish consume it. Sea turtles consume it. Even other jellyfish may feed upon it. Parasites invade its tissues, and pollution alters the waters through which it drifts. Its extraordinary reversal is a response to internal biological decline, not a shield against every external danger surrounding it. The Greenland shark faces a different vulnerability. Its immense lifespan is balanced by one of the slowest reproductive schedules known among vertebrate animals. Current evidence suggests that females may require approximately 150 years before reaching sexual maturity. Every individual, therefore, represents an enormous investment of evolutionary time. A shark lost after 100 years of life may never have reproduced even once. The disappearance of that single animal removes not only an ancient individual, but an entire lifetime of future reproductive potential that never had the opportunity to begin. The ocean quahog remembered, as Ming illustrates, how swiftly extraordinary longevity can vanish. For more than 5 centuries, the clam endured changing climates, shifting coastlines, and the rise and fall of civilizations. Its life ended within moments during an ordinary scientific preservation procedure. Researchers initially froze the specimen before realizing that the growth rings within its shell recorded the longest confirmed lifespan ever documented for an individual animal. What had survived for centuries was lost before its full significance became apparent. Scientists involved in studying the specimen later reflected publicly upon the event with considerable regret. They explained that the preservation process occurred before anyone understood the clam's exceptional age. The loss became an enduring reminder that remarkable biological discoveries can disappear through ordinary actions when their importance has not yet been recognized. In marine biology, the story of Ming is often remembered not because researchers acted carelessly, but because knowledge arrived only after the opportunity to protect the animal had already passed. The same principle extends across every organism encountered in this exploration. Biological immortality does not describe an animal that cannot die. It describes an organism whose internal aging program has been slowed, suspended, or redirected in ways uncommon among most forms of life. Cells may continue repairing themselves. Chromosomes may retain their protection. Development may even reverse toward youth. None of those remarkable abilities prevents injury, disease, predation, habitat destruction, or sudden environmental change. For millions of years, these species evolved within environments where their unusual strategies proved remarkably successful. They developed responses to natural predators, changing oceans, and the gradual pressures of evolution. What they did not encounter during most of their history were the rapid transformations introduced by industrial fishing, pollution, habitat disturbance, climate change, and expanding human activity across the seas. Evolution never prepared them for changes unfolding within only a few human generations. That contrast makes their extraordinary longevity appear even more delicate. The oldest animals in the ocean persist because their biology grants them the possibility of extraordinary time. Whether they are allowed to receive that time depends increasingly upon forces outside their own remarkable adaptations. Their greatest limitation has never been the passage of years. It has always been the world capable of ending those years long before their remarkable biology ever reaches its natural limits. There are moments when a scientific discovery does more than reveal something new. It restores an entire chapter of Earth's history that had been believed lost beyond recovery. Such a moment arrived along the Pacific coast of Canada, where the deep sea floor preserved a community that had quietly continued its existence while generations of scientists assumed it had vanished millions of years earlier. In the year 1987, researchers exploring the deep waters off the coast of British Columbia identified extensive reefs built by glass sponges. Until that discovery, the prevailing scientific view held that such reefs had disappeared approximately 40 million years ago. Fossils preserved within ancient rock formations across Europe seem to record the final chapter of these remarkable ecosystems. Instead, those fossilized remains proved to represent only part of the story. Living reefs had endured all along, hidden within deep Canadian waters beyond ordinary observation. Some of the individual glass sponges inhabiting these reefs are estimated to be thousands of years old. Generation after generation, they contributed delicate skeletal structures that gradually fused into vast living frameworks. The resulting habitat shelters, fish, crustaceans, and many other deep water organisms, creating entire ecosystems supported by growth so gradual that no human lifetime could witness more than a tiny fraction of its development. Each surviving reef represents an accumulation of biological history extending across millennia. Yet, extraordinary age provides no protection against mechanical force. One of the greatest threats comes from bottom trawling, a commercial fishing practice in which heavily weighted nets are dragged directly across the seafloor. Designed to harvest fish living near the bottom, these nets can also crush fragile habitats standing in their path. A reef requiring approximately 9,000 years of uninterrupted growth may be shattered during a single fishing pass, lasting only minutes. The contrast between creation and destruction becomes almost impossible to comprehend. What accumulated layer by layer across ages can disappear before a vessel reaches the end of its towing route. Recognition of this danger eventually led Canadian authorities to establish protected areas covering several known sponge reefs. These measures now reduce many of the most immediate risks within designated regions. Even so, protection arrived only after portions of several reefs had already sustained damage from earlier fishing activity. Conservation began not at the beginning of the story, but after irreplaceable sections of these ancient ecosystems had already been altered. The Greenland shark faces a different challenge. Rather than being deliberately targeted throughout most modern fisheries, it frequently becomes bycatch, animals unintentionally captured in nets intended for other species. Estimating the full scale of these losses remains difficult because bycatch records are not always reported with enough detail to identify every captured species accurately. Some sharks are released, yet many are already dead by the time fishing gear returns to the surface. The biological consequences extend far beyond the loss of individual animals. As explored earlier, female Greenland sharks may not reach reproductive maturity until approximately 150 years of age. Every mature adult, therefore, represents well over a century of survival before contributing to the next generation. When such an individual disappears, replacing that lost reproductive potential may require centuries rather than decades. The species is presently classified as vulnerable by the International Union for Conservation of Nature Red List. Even with that recognition, scientists still cannot determine population trends with confidence. These sharks inhabit immense depths across remote Arctic and North Atlantic waters, making comprehensive monitoring extraordinarily difficult. Their world remains challenging to study, and uncertainty itself becomes part of the conservation problem. It is difficult to protect fully what cannot yet be counted with precision. The growing understanding of these ancient animals has therefore arrived alongside an equally important realization. Their remarkable biology evolved across environments capable of supporting extraordinary spans of life. When those environments are disrupted, longevity itself loses the stable foundation upon which it depends, and the passage of thousands of years can be undone far more quickly than nature ever learned to rebuild. Some environments change so gradually that evolution can follow their pace. Generation after generation, small biological adjustments accumulate until species become finely matched to the world surrounding them. The deepest regions of the ocean belong to that category. For millions of years, they remained among the most stable habitats on Earth, allowing extraordinary forms of longevity to emerge under conditions that altered only with immense geological patience. That stability is now beginning to shift. The Arctic Ocean is warming at approximately 4 times the average rate observed across the planet as a whole. Measurements collected throughout the North Atlantic also show rising temperatures extending into deeper waters. Although those changes remain modest compared with conditions near the surface, they are unfolding within ecosystems whose inhabitants evolved under remarkably consistent thermal conditions across immense spans of time. For the Greenland shark, this change reaches into the very foundation of its biology. Its extraordinary lifespan depends upon waters remaining close to freezing throughout most of its existence. Low temperatures suppress metabolism, slowing the countless chemical reactions occurring within every living cell. When metabolism proceeds more slowly, cells divide less frequently. Fewer cellular divisions reduce the gradual shortening of chromosome ends that accompanies repeated growth and repair. As surrounding waters become warmer, metabolism increases. Increased metabolism encourages more frequent cellular division, and more frequent division gradually accelerates the processes associated with biological aging. The cold that has supported centuries of life begins to withdraw. Another challenge arrives through changing ocean chemistry. As atmospheric carbon dioxide dissolves into seawater, it lowers the acidity balance, a process known as ocean acidification. Although the ocean remains slightly alkaline overall, even relatively small shifts in this balance alter the chemical environment upon which many marine organisms depend. The effects may appear subtle at first, yet they influence biological systems refined through millions of years of remarkable stability. Glass sponges rely upon carefully regulated chemical conditions to construct their intricate skeletal frameworks from hydrated silicon compounds. Species such as Monorhaphis chuni gradually deposit these structures layer by layer across immense spans of time. Researchers continue investigating exactly how changing ocean chemistry influences this process, but evidence suggests that alterations in acidity and dissolved ions may interfere with the delicate biological mechanisms responsible for skeletal formation. An organism built through extraordinary patience depends upon equally patient environmental consistency. Ocean quahog clams face a related challenge through an entirely different material. Their shells are constructed primarily from calcium carbonate, deposited in successive growth layers that eventually preserve centuries of environmental history. Those same shells become more difficult to build and maintain as ocean acidification reduces the availability of carbonate ions required for mineral formation. The structures that protect the animal and preserve its remarkable chronological record become increasingly expensive for the organism to construct within an altered ocean. The immortal jellyfish appears more adaptable than many of the other species encountered throughout this journey. Human shipping has inadvertently transported it into distant seas through ballast water, allowing populations to spread widely across the globe. Yet even this remarkable traveler remains connected to the broader marine food web. It depends upon healthy plankton communities for nourishment, and those microscopic ecosystems are themselves changing as warming waters alter ocean circulation, seasonal productivity, and species composition. Regional declines within plankton populations eventually ripple upward through every level of marine life. Perhaps the greatest challenge lies not in the existence of change itself, but in its pace. The deep ocean has remained thermally stable across millions of years, allowing evolution to proceed gradually alongside its surroundings. The warming now being measured is occurring within only decades, an interval almost instantaneous from a geological perspective. Species adapted to extraordinary environmental consistency possess little evolutionary opportunity to respond to transformations unfolding so rapidly. These remarkable animals endured shifting continents, advancing glaciers, and ancient mass extinctions across the immense history of life on Earth. Today, many of the conditions that made such endurance possible are changing within the span of a single human lifetime, compressing geological change into decades and placing some of the planet's longest living organisms before a challenge, unlike any they have previously encountered. Knowledge does not always arrive the moment a discovery is made. Sometimes an organism is found long before the tools exist to understand what it carries. Its body becomes a collection of unanswered observations, waiting patiently for future generations of scientists to ask better questions. Many of the oldest animals on Earth now exist within that interval between discovery and understanding, preserving biological records that remain only partially translated. Every extraordinary species encountered throughout this journey can be viewed as a living library. Within its cells are molecular adaptations refined across spans of time no laboratory experiment has ever reproduced. Within its tissues are chemical signatures recording environmental histories that stretch far beyond the limits of modern scientific observation. Each surviving individual contains information gathered continuously through simply remaining alive. The Greenland shark offers a remarkable example of this hidden archive. Its genome reflects evolutionary solutions shaped through countless generations, living within one of the most stable environments on Earth. Scientists completed the first comprehensive sequencing of that genome only in the year 2024. The work marked an important beginning rather than a conclusion. Researchers are still identifying the specific genetic variations associated with exceptional longevity, efficient cellular maintenance, and resistance to accumulated biological damage. The possible medical significance of those discoveries remains an active field of investigation whose full implications have yet to emerge. The immortal jellyfish presents another scientific frontier still waiting to be fully explored. Although researchers understand that the animal can reverse its life cycle through widespread cellular reprogramming, the precise genetic networks coordinating that transformation remain incompletely mapped. Scientists continue identifying the genes that activate, suppress, and reorganize mature tissues during this extraordinary process. Each new discovery expands understanding, yet the complete sequence of molecular events has not been fully reconstructed. The phenomenon is known. Its deepest mechanisms remain only partly illuminated. Glass sponges preserve a different category of information altogether. A single ancient skeletal spicule from an individual, estimated to be approximately 11,000 years old, contains a continuous environmental record extending from the close of the last great ice age into the modern world. Layer after microscopic layer, changing ocean chemistry, became incorporated into its structure as naturally as a tree records changing seasons within its wood. Every surviving specimen therefore represents not only an individual organism, but also a chronological archive impossible to recreate once destroyed. No scientist can determine how many comparable records disappeared before anyone realized they existed. Bottom trawling altered portions of deep sponge habitats decades before their true significance became widely recognized. Some ancient individuals may have vanished without ever being documented, carrying environmental histories that ended before a single sample could be collected. Those missing chapters cannot now be recovered because the organisms that preserved them no longer exist. The story of Ming continues to illustrate the same reality from another direction. Although the clam shell still provides valuable information and remains the subject of ongoing scientific analysis, its biological future ended at the moment preservation began. Had the animal continued living, every additional year would have added another growth layer, another environmental record, another opportunity to examine how extraordinary longevity functions within a single individual across successive centuries. That unwritten continuation disappeared with the organism itself. This places science within a race that grows more urgent with each passing decade. Researchers continue discovering remarkable adaptations, while environmental change and habitat loss threaten the very organisms capable of revealing them. The challenge is no longer theoretical. Investigation and disappearance now unfold at the same time. In many cases, understanding advances more slowly than the pressures acting upon the species being studied. Yet, even beyond the possibilities of future medicines, climate records, or biological discoveries, another form of loss quietly emerges. Every one of these remarkable organisms represents an irreplaceable expression of life's capacity to explore possibilities far beyond ordinary expectation. When such a lineage disappears, the loss reaches beyond practical knowledge, carrying away a singular chapter in the long creative history through which life learned, over immense ages, what it was capable of becoming. One of the most surprising lessons offered by the ocean's longest-lived creatures is not simply that aging can proceed more slowly. It is that aging itself may not be an unavoidable law of life. Instead, it appears increasingly possible that what many animals experience is one particular evolutionary strategy among several, shaped by the demands of the environments in which they evolved rather than imposed by biology without exception. This realization has gradually reshaped the way many evolutionary biologists think about growing old. For centuries, aging was often regarded as an unavoidable consequence of living, a gradual accumulation of damage that no organism could escape. Yet the existence of species capable of maintaining healthy tissues across centuries suggests that another explanation deserves equal attention. Perhaps aging persists because, under many circumstances, it serves an evolutionary purpose. Natural selection operates upon populations rather than individuals. As environments change, organisms carrying new combinations of inherited traits may become better suited to survive than those whose genetic makeup reflects conditions that existed many generations earlier. When older individuals gradually disappear, younger generations inherit opportunities to reproduce, spreading fresh combinations of genes throughout the population. Evolution, therefore, proceeds more rapidly because successive generations replace one another within comparatively short spans of time. Viewed from this perspective, aging resembles less a biological malfunction than an adaptive process. By gradually limiting the lifespan of individuals, evolution continually refreshes the population with descendants carrying slightly different inherited characteristics. If climate shifts, food sources change, predators appear, or diseases emerge, populations capable of cycling through generations more rapidly may respond more effectively than populations dominated by exceptionally long-lived individuals whose genetic combinations originated under earlier conditions. That principle, however, depends upon change occurring often enough to reward continual replacement. In environments transformed every few seasons or every few centuries, rapid generational turnover may provide a genuine evolutionary advantage. The benefits of producing descendants with new combinations of inherited traits outweigh the advantages of preserving exceptionally old individuals whose biology reflects an earlier world. The deep ocean presents a remarkably different circumstance. Across immense geological intervals, its temperature, pressure, chemistry, and available resources have altered with extraordinary slowness. Conditions certainly changed over millions of years, yet those changes unfolded gradually, compared with the dynamic environments experienced across coastlines, rivers, forests, and grasslands. Under such remarkable stability, the evolutionary value of replacing healthy individuals simply becomes less compelling. An organism, already well suited to this enduring environment, gains little advantage from disappearing merely to create space for descendants carrying slightly altered genetic combinations. If the surrounding world remains broadly similar across generation after generation, preserving an individual capable of maintaining healthy tissues for centuries may become every bit as successful as replacing it rapidly. Natural selection therefore favors a different balance, investing more heavily in maintenance, repair, and continued survival rather than accelerated turnover. The remarkable creatures encountered throughout this journey illustrate precisely that possibility. They did not abolish aging through a single shared innovation. Instead, each lineage discovered its own biological pathway toward prolonged maintenance while inhabiting an environment where extended persistence remained evolutionarily worthwhile. The deep ocean did not intentionally produce immortal organisms. Rather, it removed many of the selective pressures that normally reward shorter lives, allowing alternative strategies to emerge through countless generations. Within this framework, aging itself appears less like a universal destiny and more like a solution to a particular evolutionary challenge. It helps populations adapt efficiently when surroundings continually demand new biological responses. Where those demands diminish, the necessity for rapid replacement also weakens, opening space for organisms capable of extraordinary endurance to flourish. Seen in that light, the oldest animals beneath the sea do more than expand the known limits of lifespan. They reveal that aging is neither identical across all living things nor equally valuable within every environment. Their existence suggests that evolution has always possessed more than one answer to the question of how long a life may continue, and that the path ultimately taken depends as much upon the world surrounding an organism as upon the cells that compose its body. The search to understand aging has entered a different era. For much of modern biology, researchers focused on describing the gradual changes that accompany growing older, carefully documenting declining cellular function, accumulating molecular damage, and the diseases that emerge more frequently with advancing age. Increasingly, however, the field known as geroscience has begun pursuing a different objective. Rather than asking only how aging occurs, scientists are investigating whether specific biological processes that contribute to aging can be slowed, modified, or, in carefully limited ways, repaired. This shift has drawn growing attention towards some of the longest-lived animals in the ocean. Their value does not lie in providing direct blueprints for human biology. Humans will never become Greenland sharks or immortal jellyfish. Instead, these remarkable organisms reveal molecular strategies that evolution has already tested across immense spans of time. Researchers hope that understanding those strategies may uncover biological pathways already present within human cells, pathways that might someday be activated through carefully designed medicines. The Greenland shark has become one of the most compelling examples. Following the publication of its comprehensive genome during the year 2024, researchers identified multiple duplicated genes involved in repairing damaged deoxyribonucleic acid and maintaining cellular stability. Scientists are not attempting to transfer shark genes into people. The goal is far more precise. By identifying the molecular pathways strengthened within the shark, pharmaceutical researchers hope to develop small molecule medicines capable of stimulating comparable repair mechanisms already present within human cells. Investigators at the University of Copenhagen have described this possibility directly. They noted that the duplicated repair genes observed within the Greenland shark highlight specific molecular targets worthy of further investigation. If equivalent pathways can be activated safely in human tissues, they may reduce the gradual accumulation of deoxyribonucleic acid damage that accompanies aging. Such an approach remains experimental, yet it illustrates how studying an ancient marine animal can influence entirely new directions in medical research. The immortal jellyfish offers a different opportunity. Its remarkable ability to reorganize mature cells into entirely new developmental pathways has attracted considerable attention from scientists studying regenerative medicine. Researchers continue examining the genetic networks responsible for this transformation in the hope of understanding how controlled cellular reprogramming might someday assist damaged human tissues. Potential applications under investigation include replacing neurons lost through neurodegenerative disease, encouraging regeneration of heart muscle following cardiac injury, and restoring certain tissues affected by age-related degeneration. These possibilities remain subjects of ongoing laboratory research rather than established medical treatments. Evidence gathered from other long-lived marine organisms also contributes to this expanding field. Studies examining telomerase activity within exceptionally long-lived species, including glass sponges and ocean quahog clams, are improving scientific understanding of how chromosome protection can continue for extended periods without immediately producing uncontrolled cellular growth. One of the greatest challenges in human medicine is that broadly increasing telomerase activity may also increase the risk of cancer. Researchers therefore seek methods capable of activating these protective mechanisms only within carefully selected tissues and only under tightly controlled conditions. Every one of these investigations remains within the research stage. None has produced an approved therapy capable of extending healthy human lifespan through the mechanisms explored in these remarkable marine species. Progress requires years of careful laboratory testing, clinical evaluation, and safety assessment before any discovery can become practical medicine. Biology rarely yields its deepest lessons quickly, particularly when those lessons concern processes as fundamental as aging itself. Yet a remarkable pattern has emerged across all of these scientific efforts. Each began not with an invention created inside a laboratory, but with careful observation of an organism already solving a biological challenge in its own way beneath the ocean. Long before human science recognized these possibilities, evolution had already explored them through countless generations. The deepest waters became an immense natural laboratory, preserving experiments conducted across millions of years, and only now are researchers beginning to understand the questions those extraordinary lives have been answering all along. Human history is often measured through generations. A grandparent remembers one world, a parent another, and a child inherits a third. Within only a handful of lives, languages shift, borders move, technologies emerge, and entire civilizations transform. Against that familiar rhythm, the lifespan of the Greenland shark introduces a scale of existence that resists ordinary intuition. A Greenland shark, estimated to be about 400 years old, experiences the passage of biological time at a remarkably different pace from a human being. Its slow metabolism, gradual growth, and delayed reproduction stretch the arc of life across centuries rather than decades. If the proportions of its lifespan could be translated into human experience, what people commonly think of as an entire lifetime would occupy only a comparatively early chapter in the shark's own biological journey. The span from human birth through education, adulthood, family life, and old age would correspond only to something resembling adolescence within the rhythm of this extraordinary animal. Such comparisons are only approximations, yet they help illuminate how differently longevity can be expressed across living organisms. Time itself passes equally for every creature, but biology determines how quickly life unfolds within that shared passage. The Greenland shark does not receive more hours than any other animal. Instead, it distributes the milestones of existence across an interval so extended that familiar human expectations begin to lose their meaning. A shark approaching 4 centuries of age would have entered the world around the early decades of the 17th century, during a period when much of modern history still lay in the future. It would already have been swimming beneath Arctic waters before many nations assumed their present forms, before the great advances of modern medicine, before electricity transformed cities, before powered flight, before space exploration, before computers, and before communication spread across the planet at the speed of light. Entire political systems have risen and disappeared, while that same individual continued its gradual movement through the depths. None of these transformations unfolded within the shark's awareness as historical events. It possesses no understanding of empires, discoveries, revolutions, or calendars. Its nervous system does not construct narratives about passing centuries or measure existence through anniversaries. The succession of human history has occurred almost entirely beyond its perception. While societies above the ocean repeatedly reinvented themselves, the shark continued following the biological rhythms that shaped its species long before human civilization emerged. This contrast reshapes the human sense of duration. A scientist may devote an entire professional career to understanding Greenland sharks, beginning as a student, continuing through decades of research, and eventually passing that knowledge to younger colleagues. From the perspective of the shark itself, that entire scientific career represents only a brief interval within its own lifespan. Multiple generations of researchers may study the same individual without that animal ever approaching the natural conclusion of its biological existence. The realization carries an unusual form of perspective. Human history often feels immense because each generation experiences only a small portion of it directly. Yet for a Greenland shark, the sequence of events that humans describe as modern civilization occupies only one extended phase within a life still capable of continuing. Long after today's laboratories have changed, today's scientific debates have evolved, and today's observers have themselves become part of history, the same shark may still inhabit the cold northern waters where its journey began centuries earlier. The Greenland shark does not witness history in the human sense, yet its continued existence quietly bridges eras that people ordinarily separate into distant chapters. Within a single living body, centuries become continuous rather than divided. Measured against that extraordinary scale, even the longest human life appears remarkably brief, not diminished in meaning, but compressed into a fleeting interval beside an organism whose biological clock advances with astonishing patience, preparing the mind for forms of longevity that extend even further beyond familiar experience. Imagine a life that began before the first cities existed, before fields were planted in ordered rows, before writing preserved memory beyond the spoken word. Deep upon the sea floor, one remarkable glass sponge may have begun growing during a moment when the planet itself was completing one of its greatest climatic transformations. Its existence reaches backward, not merely through recorded history, but into a world that survives today only through geology, archaeology, and fragments of ancient ice. An individual estimated to be approximately 11,000 years old would have attached itself to the seabed near the close of the Younger Dryas, the final major return of glacial conditions before the present warm interval became established. As that cold period ended, global temperatures rose rapidly across geological timescales, reshaping coastlines, ecosystems, and weather patterns into forms more recognizable today. The sponge remained where it had settled, while the world above entered an entirely different age. At that distant moment, the landscape, now known as the British Isles, was still connected to continental Europe by broad stretches of exposed land. Sea levels stood many meters lower than they do today. That is nearly many tens of feet lower, because enormous volumes of water remained locked within continental ice sheets. Across northern Africa, the Sahara was gradually entering a wetter climatic period, during which rivers, lakes, and grasslands spread across regions now dominated by desert. Human communities lived as hunter-gatherers, relying upon carefully crafted stone tools while adapting to changing environments that still carried the lingering influence of the Great Ice Age. Across the thousands of years that followed, humanity transformed its relationship with the world. Agriculture emerged, allowing permanent settlements to expand into villages and cities. Written language preserved ideas beyond living memory. Mathematics provided a new language for describing patterns. Philosophy examined existence itself. Religious traditions shaped civilizations. Medicine slowly separated observation from superstition. Systems of law organized increasingly complex societies. Architecture lifted stone into monuments that survived generations. Navigation connected distant continents. Chemistry and physics revealed hidden principles governing matter and energy. Eventually, technological innovation became sophisticated enough to descend into the deep ocean and estimate the age of the extraordinary organism that had remained in one place throughout nearly all of it. The sponge itself experienced none of these achievements as events unfolding through time. Unlike animals possessing brains capable of constructing memories or anticipating the future, a glass sponge has no nervous system through which history acquires personal meaning. It does not remember the passing of centuries, because memory, in the human sense, does not exist within its biology. It does not anticipate tomorrow or reflect upon yesterday. For such an organism, existence is not measured through remembered moments, but through uninterrupted continuation. This distinction reshapes the meaning of longevity. Human beings naturally associate long life with accumulated experience, believing that additional years produce deeper memories, broader knowledge, and richer personal histories. The oldest glass sponge represents something entirely different. Its extraordinary age is expressed not through conscious recollection, but through persistent biological continuity. It has remained present while civilizations appeared, flourished, transformed, and disappeared without ever recognizing that such changes occurred. Yet this absence of awareness does not diminish its scientific significance. On the contrary, it elevates it. Through its gradual growth, the sponge preserved chemical information about the surrounding ocean across thousands of years with remarkable consistency. Without intending to record anything, it became one of the longest continuous biological archives ever produced by life itself, preserving environmental history through its own existence rather than through observation. Perhaps that is why such extraordinary longevity remains difficult to comprehend. Human thought instinctively links time with experience, while the glass sponge separates the two completely. Its life demonstrates that duration and awareness are not identical qualities. One may continue almost beyond imagination without possessing any understanding of the passage itself, becoming not a storyteller of history, but its enduring biological record, existing through ages that unfolded entirely beyond the reach of conscious experience. Scientific knowledge has remarkable reach. It can reveal the structure of a gene, trace the movement of atoms through living tissue, estimate the age of an organism from the chemistry preserved within its body, and describe the molecular pathways that allow life to endure across astonishing spans of time. Yet beyond those achievements lies a boundary that every scientific discipline eventually encounters, a place where measurement remains possible but meaning becomes far more difficult to define. The extraordinary animals encountered throughout this journey bring that boundary into unusually clear focus. Researchers can estimate the lifespan of a Greenland shark, sequence its genome, identify duplicated repair genes, and investigate the biological systems that preserve its cells across centuries. Each year, new discoveries refine that understanding. The mechanisms become more detailed, the evidence more complete, and the picture more precise. Even so, one question remains beyond the reach of instruments and laboratory analysis. Science can explain how such longevity exists. It cannot determine what that longevity is like for the organism itself. Everything presently understood about the Greenland shark suggests that it possesses no capacity for the kind of self-aware awareness through which human beings perceive the passage of decades. It does not recognize anniversaries or compare its present existence with distant memories spanning centuries. It does not know that it has become exceptionally old. It does not understand that generations of human observers have come and gone while its own life has continued. The immense duration of its existence remains biologically real without necessarily becoming psychologically meaningful. The glass sponge carries this separation even further. Unlike animals possessing brains or centralized nervous systems, it has no anatomical structure capable of assembling experiences into a continuous narrative. Thousands of years may pass while its tissues continue growing, yet there is no known mechanism through which those years accumulate as remembered time. Continuation occurs, but not recollection. Persistence exists, but not autobiography. The organism remains alive without constructing an internal story about its own existence. The immortal jellyfish offers another variation on the same theme. When environmental stress triggers its remarkable return to an earlier stage of development, the transformation unfolds through a cascade of biochemical processes shaped by evolution. There is no evidence that the animal chooses this transition or understands what is happening as its body reorganizes. The restart of its life cycle represents a physiological response rather than a conscious decision. Biology accomplishes something extraordinary without requiring awareness of the transformation itself. Taken together, these organisms reveal an unexpected pattern. The longest-lived creatures presently known are also among those least likely to possess any awareness of their extraordinary longevity. Their survival depends upon efficient biological systems rather than reflective consciousness. They continue because their bodies successfully maintain themselves, not because they contemplate the passage of time or assign personal meaning to their endurance. This observation also illuminates another aspect of evolution. Consciousness is biologically demanding. Nervous systems require considerable energy to construct, maintain, and operate. Complex perception, memory, learning, and decision-making all carry substantial metabolic costs. They offer immense advantages in dynamic environments where rapid behavioral adaptation improves survival. Yet those same advantages are not equally necessary within every ecological setting. Some organisms prosper through remarkable simplicity, investing their biological resources in persistence rather than perception. Perhaps that is why human beings experience time so differently from the oldest inhabitants of the deep ocean. For people, the passing of years becomes inseparable from memory, expectation, joy, regret, anticipation, and reflection. Duration acquires emotional texture because consciousness continually compares what has been with what may still come. The ancient organisms beneath the sea reveal another possibility altogether, one in which life extends almost beyond imagination while remaining free from the psychological weight that humans attach to every passing year, reminding us that longevity and the experience of time are not necessarily the same phenomenon. Every species carries the imprint of the world that shaped it. A lifespan is not determined by biology alone, but by the countless environmental pressures that gradually favored one survival strategy over another. Seen from this perspective, the human lifespan is neither unusually short nor unusually long. It is the result of an evolutionary balance between energy, reproduction, adaptation, and the remarkable complexity of the human body. For most of human evolutionary history, life unfolded within landscapes defined by uncertainty. Food availability fluctuated with changing seasons. Predators remained an ever-present danger. Diseases continually reshaped populations. Climate shifted over generations rather than geological ages, demanding flexibility rather than permanence. Under such conditions, producing offspring capable of adapting to new circumstances often provided greater evolutionary success than preserving individual bodies for many additional centuries. Human biology, therefore, invested heavily in versatility instead of extraordinary longevity. Muscles capable of sustained movement, immune systems prepared for diverse pathogens, intricate hormonal regulation, rapid wound healing, and an exceptionally sophisticated nervous system all require immense and continuous energy. Among these investments, none is more demanding than the human brain. Although representing only a modest fraction of total body mass, the adult human brain consumes approximately 1/5 of the body's available energy during ordinary activity. That constant demand supports memory, language, imagination, planning, emotional awareness, abstract reasoning, and the ability to learn throughout life. Every conversation, invention, work of art, scientific discovery, and technological achievement depends upon this extraordinarily expensive organ remaining active from moment to moment. Maintaining such complexity across spans approaching many centuries would require biological compromises unlike anything found within the human lineage. Either energy intake would need to remain remarkably stable over immense intervals, or other physiological systems would require substantial reduction to support the continuing metabolic cost. Evolution instead favored a different solution, one emphasizing intelligence, behavioral flexibility, and cultural adaptation over extreme biological persistence. The oldest inhabitants of the deep ocean illustrate the opposite strategy. A glass sponge can continue growing for astonishing lengths of time precisely because it invests almost nothing in the structures that consume so much energy in more complex animals. It possesses no brain, no centralized nervous system, no capacity for learning, no deliberate movement through its surroundings, and no need to process an endless stream of sensory information. Its existence follows an elegant biological economy. It remains attached to the seafloor, filters surrounding water, repairs itself, and continues. The Greenland shark occupies a different position along the same spectrum. Although vastly more complex than a sponge, its nervous system remains comparatively simple beside that of mammals. It moves gradually, feeds infrequently, matures over extraordinary spans of time, and inhabits an environment where relatively few predators threaten fully grown individuals. Much of its biology favors conservation rather than constant activity. Energy that might otherwise support rapid growth or elaborate behavior instead contributes to long-term maintenance. Human longevity is therefore constrained by more than chromosome biology or the gradual shortening of protective chromosome ends. It is also shaped by the energetic cost of sustaining everything that defines human existence. The abilities to communicate through language, preserve knowledge across generations, imagine possible futures, create civilizations, compose music, develop mathematics, and eventually descend into the deep ocean to study these remarkable organisms all emerge from biological machinery that is exceptionally expensive to maintain. A striking pattern gradually comes into view. The species capable of surviving for the greatest lengths of time are generally those that invested less in conscious complexity, while humans followed an evolutionary path that exchanged extraordinary persistence for extraordinary awareness. One strategy preserves the body across immense stretches of time. The other transforms comparatively brief lives into lives rich with memory, imagination, curiosity, and discovery. In that contrast, the oldest creatures beneath the sea reveal that longevity is only one possible measure of biological success, and that evolution has always balanced endurance against the remarkable capacities that make a life consciously experienced. Among the oldest living organisms on Earth, there is an unexpected absence of struggle. Their extraordinary longevity does not arise from relentless competition against time or from any deliberate effort to resist the gradual passage of years. Instead, it emerges from biological processes that unfold without intention, following patterns established through evolution long before consciousness entered the story. The Greenland shark offers perhaps the clearest example. Nothing in its behavior suggests resistance against aging as humans understand the idea. It does not accelerate when danger is absent, nor does it expend unnecessary energy searching for constant opportunities. Its body has become exquisitely matched to an environment where patience carries little cost. Every movement conserves resources. Every physiological process proceeds at a measured pace. The remarkable lifespan that follows is not the result of determination, but of a biology that rarely demands haste. The same principle appears in an even simpler form within the glass sponge, anchored to the seafloor. It continues the same modest activity through season after season and millennium after millennium. Water passes through its body. Nutrients are gathered. New layers of silica become part of its delicate framework. Growth continues almost imperceptibly, not because the organism strives to preserve itself, but because its ordinary existence already provides everything required for extraordinary persistence. Longevity emerges from repetition rather than ambition. The immortal jellyfish reveals another variation of this pattern. When environmental stress activates the remarkable return to an earlier stage of development, no decision has been made. No instinct weighs alternatives before selecting a path. A biochemical sequence unfolds because countless generations of evolution preserved that response. The restarting of the life cycle is neither victory nor escape. It is simply the continuation of a molecular program that has remained effective across immense stretches of evolutionary history. Sea anemones illustrate the same principle through their capacity to divide and produce genetically identical descendants. They do not seek permanence as an abstract goal. Cellular processes proceed according to inherited biological instructions, and persistence follows naturally from those ordinary functions. What appears extraordinary from a human perspective exists only because the organism continues behaving exactly as its biology has always directed. Taken together, these animals reveal a striking contrast with humanity. Across laboratories, universities, hospitals, and research centers, enormous effort is devoted to understanding the causes of aging and discovering methods that might delay its effects. Scientists examine genes, proteins, chromosomes, metabolism, and cellular repair with extraordinary care, hoping to preserve healthy life for longer periods. Meanwhile, beneath the ocean, organisms displaying many of those desired characteristics continue existing without any awareness that their biology has become an object of intense scientific fascination. Perhaps the greatest difference lies not in lifespan itself, but in the relationship each organism has with mortality. Human beings recognize that life is finite. Memory allows the past to accumulate. Imagination projects possibilities into the future, and consciousness continually measures the distance between them. The oldest marine organisms possess no evidence of such reflection. Their lives unfold without contemplating beginnings or endings. When death arrives, it generally comes through injury, environmental disruption, predation, or other forces originating beyond the ordinary maintenance of their own bodies. In that contrast, a deeper pattern gradually becomes visible. Evolution produced organisms that persist because their biology remains remarkably stable, not because they possess an understanding of survival. The most enduring lives beneath the sea are carried forward by mechanisms rather than intentions, by continuity rather than aspiration. Human beings, by comparison, experience longevity through awareness, attaching meaning to every passing year. The oldest creatures in the ocean demonstrate another possibility altogether, one in which existence continues with extraordinary endurance, untouched by the ideas of mortality that occupy so much of the human imagination. After following these remarkable organisms across such immense stretches of biological time, another thought gradually emerges. It no longer concerns how these animals avoid aging, nor how researchers investigate their extraordinary biology. Instead, attention turns toward a different idea, one that slowly forms as every discovery is placed beside the next. If aging is not universal, then what purpose does it ordinarily serve for the creatures that experience it? Across most of evolutionary history, aging appears to have been less a mistake than a consequence of ecological circumstance. Organisms lived in environments where food supplies changed, climates shifted, predators remained abundant, and diseases continually altered the balance of survival. Under those conditions, producing new generations capable of inheriting fresh combinations of genetic variation often proved more valuable than preserving older individuals indefinitely. Evolution favored populations that could adapt, even when individual lives remained comparatively brief. That perspective transforms aging from an unavoidable biological law into one possible solution among many. Earlier, the remarkable inhabitants of the deep ocean revealed another solution altogether, one shaped by environments where stability endured across immense spans of time. In those settings, maintaining an existing body could become as successful as replacing it. The biological clock, therefore, was never fixed in the first place. It changed according to the demands imposed by each environment. Human beings occupy an unusual position within this story. Unlike every species that came before, people have gradually learned enough about genetics, cellular maintenance, metabolism, and evolutionary biology to recognize that aging itself has underlying mechanisms open to scientific investigation. Those mechanisms are no longer hidden. Laboratories throughout the world continue identifying the pathways through which cells repair damage, regulate growth, and preserve function across time. For the first time in Earth's history, one species has become capable of asking whether those mechanisms might eventually be adjusted deliberately rather than merely inherited. The remarkable animals encountered throughout this journey should not be understood as promises of human immortality. Their value lies elsewhere. They demonstrate that life can follow evolutionary paths in which extraordinary longevity emerges naturally when particular ecological pressures disappear or become greatly reduced. They reveal that biology contains possibilities extending well beyond what human experience alone might suggest. In doing so, they replace certainty with a broader understanding of what evolution is capable of producing. Determining how humanity should respond to that knowledge belongs to a different kind of conversation. Scientific investigation can describe genes, proteins, cells, and molecular pathways with increasing precision. It can explain why some organisms persist for astonishing intervals while others do not. Deciding whether such knowledge ought to reshape the future of human aging reaches beyond experimental evidence alone. It touches philosophy, ethics, culture, and the values through which societies understand the meaning of a human life. The Greenland shark possesses no awareness that its existence has already extended across approximately 4 centuries. The glass sponge has no understanding that its life began near the close of the last great cold interval before the modern climate emerged. The immortal jellyfish does not recognize that its unusual life cycle has become one of biology's most fascinating discoveries. None pursued longevity as an ambition. Each simply inherited a successful strategy refined through countless generations by evolutionary change unfolding across millions of years. Human beings arrived at this moment by a different path. Biological evolution provided the capacity for language, memory, curiosity, and cumulative learning. Culture carried those abilities forward, allowing each generation to preserve discoveries for the next instead of beginning again. Through that growing body of knowledge, humanity eventually descended into the deep ocean and uncovered organisms that had been following their ancient patterns long before civilization itself appeared. Those discoveries now carry another responsibility as well, because the oldest living archives beneath the sea continue existing while the changing world above increasingly threatens the conditions that allowed them to endure. The ocean has conducted these biological experiments in darkness, beyond ordinary notice, for far longer than human civilization has existed, and only recently has humanity begun learning how to read what they have patiently preserved. Long before human beings developed science, before language preserved knowledge across generations, and before curiosity could be transformed into careful observation, the ocean had already begun conducting its own vast biological trials. No intention guided them, no prediction shaped their outcome. They unfolded through the gradual accumulation of countless tiny variations, each tested against changing seas over spans of time so immense that even the history of civilization occupies only a fleeting interval by comparison. Among the oldest participants in those ancient processes are the glass sponges. Their lineage reaches back to the Cambrian diversification of animal life, when multicellular organisms rapidly expanded into many of the fundamental body forms from which later animal evolution emerged. Across hundreds of millions of years, continents drifted, oceans opened and closed, climates warmed and cooled, and entire branches of life disappeared. Through all of those transformations, descendants of those early sponges continued refining a remarkably economical way of living, one that required little movement, little energy, and extraordinary persistence. The Greenland shark belongs to another lineage of remarkable endurance. Its ancestors were already swimming through ancient seas at least 200 million years ago. During that immense interval, the planet experienced repeated environmental upheavals unlike anything recorded within human history. The Triassic-Jurassic extinction reshaped ecosystems across land and sea. Much later, the Cretaceous-Paleogene extinction ended the age of the non-avian dinosaurs while transforming marine communities throughout the world. Numerous additional extinction events altered ocean life before, between, and after those famous chapters. Yet the lineage leading to the Greenland shark continued through them all. Their persistence did not arise from unusually advanced intelligence or elaborate social behavior. They built no shelters, developed no tools, and passed no accumulated knowledge from one generation to the next. Instead, their success rested upon biological designs remarkably well suited to stable marine environments. Slow growth, restrained energy use, efficient maintenance of living tissues, and resilience under demanding conditions repeatedly proved sufficient. Evolution preserved those characteristics because, across immense intervals, they continued working. From a human perspective, scientific research begins with a hypothesis, proceeds through observation, and arrives at conclusions supported by evidence. Evolution follows no such structure. It has no destination and no awareness of its own outcomes. Every successful organism simply represents one arrangement of biology that continued functioning while countless alternatives disappeared. Across geological time, the deep ocean became an immense arena in which those arrangements were tested continuously, generation after generation, without pause. Only recently has humanity begun recognizing the significance of the results. Beneath the surface today, organisms continue living whose ancestors survived events separated by millions of years. Some individual glass sponges living now first attach themselves to the seafloor near the close of the last great ice age and have remained there ever since. Other ancient marine organisms preserve biological strategies whose origins extend so deeply into Earth's history that reconstructing their evolutionary pathways remains an ongoing scientific challenge. Their cells, genes, and tissues contain information that researchers are only beginning to understand, asking questions that earlier generations could scarcely have imagined. Perhaps the greatest surprise is how much remains unread. Every newly sequenced genome, every carefully analyzed tissue sample, and every expedition into the deep sea reveals additional complexity rather than final answers. These organisms are not merely unusual survivors. They are living records of evolutionary solutions refined across spans of time beyond ordinary comprehension, carrying biological knowledge preserved not in books or monuments, but within continuously functioning bodies. For many centuries, humanity searched mountaintops, deserts, caves, and ancient ruins for the oldest witnesses to Earth's past. Yet some of the most enduring archives were never standing beneath open skies at all. They remained beneath the surface, beyond ordinary reach, continuing lives measured in centuries and millennia, while history unfolded above them. Today they remain there still, holding biological stories written across evolutionary time, increasingly accessible to human exploration, and increasingly vulnerable to the same species that has only just begun learning how extraordinary they truly are. At this very moment, while ordinary human life continues beneath changing skies, the oldest living chapters of Earth's biological history remain exactly where they have long belonged. They are not preserved behind glass, cataloged within libraries, or displayed inside museums. They continue living, carrying on the same ordinary existence that has sustained them across spans of time almost impossible to imagine. Somewhere on the floor of the Indo-Pacific, a glass sponge that first attached itself to the seafloor near the end of the last great ice age continues filtering seawater through its intricate framework of silica. Each passing current brings microscopic nourishment. Each day adds only the slightest continuation to a structure that has grown with extraordinary patience across thousands of years. There is no awareness of history unfolding far above. There is only the uninterrupted rhythm of a body continuing exactly as evolution shaped it to continue. Far to the north, beneath waters that remain close to freezing, a Greenland shark born before the United States came into existence glides through the North Atlantic. Its movements remain unhurried, because nothing within its biology requires haste. The same measured pace that once carried it through oceans known only to sailing vessels now carries it through seas crossed by satellites, research ships, and underwater vehicles. If no injury, fishing activity, or environmental disruption interrupts that journey, many more decades, perhaps even centuries, may still belong to that single animal. Elsewhere, within warmer currents carried across the open sea, an immortal jellyfish, no larger than a human fingernail, drifts almost unnoticed among countless other small marine organisms. Should severe stress damage its body, a familiar biochemical sequence may begin once again. Cells reorganize according to ancient developmental pathways, gradually returning the organism to an earlier stage of its life cycle. Within approximately 72 hours, that remarkable transformation can be completed, not as an act of intention, but as another ordinary expression of inherited biology. These are not legends carried forward through generations of storytelling. They are present realities unfolding continuously beneath the ocean surface. At this very moment, organisms possessing extraordinary relationships with time continue existing in places that remain largely beyond direct human observation. Many inhabit depths where pressure reaches hundreds of atmospheres, where sunlight never arrives, and where conditions have persisted with remarkable stability across intervals longer than recorded human history. Nothing about their behavior suggests that they recognize their own uniqueness. The glass sponge does not know that scientists estimate its age through careful analysis. The Greenland shark does not understand that its genome has become the subject of international research. The immortal jellyfish possesses no awareness that its unusual life cycle has inspired laboratories around the world to investigate the biology of cellular renewal. Their existence remains entirely independent of the attention now directed toward them. Yet another truth accompanies that realization. The same species that has finally learned how unusual these organisms truly are also possesses an unprecedented capacity to alter the environments that allowed them to endure. Warming oceans, changing chemistry, habitat disturbance, pollution, and intensive exploitation increasingly reach ecosystems that once remained comparatively stable across geological time. The biological archives, preserved beneath the sea, continue their ancient rhythms, even as the conditions surrounding them become less certain with each passing generation. For countless millennia, the ocean sheltered these remarkable experiments in longevity beneath immense depths and immense spans of time. Only recently has humanity become capable of descending far enough, observing carefully enough, and understanding deeply enough to recognize what has always been there. Even now, while these words are heard, those ancient lives continue beyond ordinary sight, following patterns older than civilization itself. They remain among the oldest living witnesses on Earth, still continuing their patient existence beneath the surface, while the rapidly changing world above increasingly reaches down toward them, leaving only one final reflection before this journey comes to its close. The longest living inhabitants of our planet receive almost none of the recognition that human history so readily gives to monuments, rulers, discoveries, or civilizations. Their names exist because science eventually encountered them and placed words upon lives that had already been unfolding for ages. Before that moment, they simply existed beyond notice, continuing their ordinary patterns beneath the sea while generations rose, flourished, and disappeared above the surface. During those immense intervals, every human achievement remained temporary. Kingdoms expanded before fading into memory. Languages emerged, transformed, and vanished. Belief systems reshaped societies before yielding to those that followed. Governments formed, dissolved, and formed again. Cities appeared where wilderness once stood, only to become ruins beneath later civilizations. Through every chapter of that unfolding story, the oldest organisms beneath the ocean remained untouched by the ideas that defined human history. Their persistence did not come from extraordinary strength. They did not dominate their surroundings through speed, aggression, or innovation. They did not invent tools, organize societies, or alter the world to suit their needs. Instead, evolution shaped them toward another kind of endurance. Their bodies required little. Their environments changed slowly. Their biological systems favored maintenance over haste, efficiency over excess, and continuity over constant transformation. For countless generations, that proved to be enough. For much of human existence, those distant habitats remained beyond practical reach. The great depths protected these ancient organisms not through barriers deliberately placed around them, but simply through remoteness, darkness, immense pressure, and vast distances discouraged exploration long before technology could overcome them. Those same conditions also preserved ways of living that had remained remarkably stable across geological time. That separation is becoming increasingly fragile. Scientific exploration now reaches places once considered inaccessible, revealing organisms whose lives stretch across centuries and millennia. At the same time, warming oceans, habitat disturbance, pollution, and expanding human activity increasingly influence regions that for immense spans of time changed only gradually. The oldest living archives beneath the sea are no longer distant from human history. They have become part of it. Beyond their importance for medicine, beyond the climate records preserved within their tissues, and beyond every possible scientific application, these remarkable organisms offer something less tangible but equally valuable. They reveal another relationship with time itself. Human beings measure existence through calendars, birthdays, anniversaries, memories, expectations, and the awareness that each passing year carries lasting significance. The oldest creatures beneath the sea participate in none of those ideas. A glass sponge that has remained alive for approximately 11,000 years possesses no understanding of what a single year represents. A Greenland shark living across 4 centuries has no concept of centuries at all. An immortal jellyfish returning repeatedly through its unusual developmental cycle carries no awareness that its biological history has become extraordinary in human eyes. Their longevity does not arise because they somehow stand above time. Rather, they continue because nothing within their biology requires them to divide existence into remembered moments and anticipated futures. Perhaps that is the gentlest lesson carried by these ancient lives. Endurance does not always announce itself through dramatic change. Sometimes it appears as uninterrupted continuation, sustained by ordinary biological processes repeated across intervals so vast that human imagination struggles to contain them. The oldest living organisms beneath the sea have followed that path since long before the first cities, the first written histories, or the first maps of the world. Even now they remain there, beneath waters that still conceal far more than they reveal, continuing lives that began before much of human history had even started. Their existence offers no final answer and no complete resolution. It leaves only the enduring presence of an ocean older than civilization, deeper than memory, and still holding countless biological stories that humanity has only just begun to notice. Among those unseen depths, the oldest living creatures on Earth continue their patient existence, and for now, they are still alive.